Hello friend, welcome to the channel Pharmacy Dictionary. There are 4 parameters under pharmacokinetic. These are ADME, and just now we have completed the all points regarding absorption. Now it's time to understand the basic concept and important phenomena under the D, that means distribution. So today's topic is pharmacokinetic and the distribution, along with the important consequence and important point regarding plasma protein binding. So what is the plasma protein binding? What is the distribution? We are going to study all about this topic today. So first of all, what is the meaning of distribution? Whenever any patient took any kind of medicine via any route of drug administration, like if this is a solid dosage form, so I am going to take this medicine via oral route of drug administration. After reaching through the stomach, that drug will go for disintegration and dissolution, and then that will go for the absorption. What is the absorption? Movement of drug from its site of administration toward the systemic circulation or blood vessel, that is called absorption. So what is going to happen in the next sequence? What will be happen after absorption? Should that drug remain on the blood on the same portion all the time? No, that is not possible, and that should not be happen too. So what is going to happen? This drug which has been absorbed by the blood vessel now will start moving throughout the body, and I can say that throughout the cell present in the body, and that movement of drug from it the absorption side, that means blood, throughout the all cells inside body, this is called distribution. Actually, in general terms, in Hindi we used to say baantna. That means if we have something much in amount, then we should distribute it to all the particular persons in the house or in the particular conference which is going to take place. That means to simply distribute that particular object through all of the particular participant in the case. Or what is the distribution in case of biological phenomena? If some tissue is having much concentration of any drug, that may be converted into a toxic effect. So that tissue try to make the equilibrium between all the surrounding tissue, so that each and every part of our body will receive the drug and produce their desirable action once they will occupy their binding site, that means receptor. So in general sense, we can say distribution is a movement of drug from blood, because the drug is absorbed in blood only. So movement of drug from the blood to all other tissue in the body is simply called distribution. So just give you the example, I am having 10 number of toffees. These are 10 toffees. So what I am going to do that is, if I am about to distribute this toffee to all the students available in my coaching classes, or just all the children available in my colony, so what are the options with me? I have many options. First option says this is 10 chocolates, so I should distribute every student a single chocolate, so that all may be satisfied. But I have another option that I will distribute out of this 10 5 chocolate, and I will keep 5 chocolate with me. So I am not going to distribute this chocolate. So how many students will receive this drug or this chocolate? Only 5 students. Or what about the 5 which is having with me? That this 5 chocolate will not going to be distributed in the circulation system, and that will remain unavailable to the children or to the student until I will release one by one, then only they will have. So this is the important phenomena of distribution, that drug should be distributed in an equilibrium manner, so that there will be a positive homeogenesis condition. So coming to the next point under distribution, second point say it should cross the several barrier before reaching to the site of action. There are so many biological barriers we have seen in case of absorption, and to achieve the distribution manner, to distribute the drug throughout the body, that particular drug should cross different biological barrier like blood-brain barrier, blood placental barrier, blood testis barrier, so that they will reach their site of action accordingly to their lipid or water solubility. These 3 barriers which I have explained, like blood-brain barrier, blood testis barrier and blood placental barrier, are very specific barrier. Specifically, blood-brain barrier is a tight junction of cell. They allow only those drugs which are highly lipid soluble drug. Water soluble drug cannot cross through blood-brain barrier, and the movement process of drug from the particular site of absorption to the distribution site is almost same as in case of absorption, like passive diffusion and endocytosis, or we can say endocytosis, or cell mediated transport, or all other parameters which are equal to the absorption parameter. So what are the basic factors which may affect, which may govern the rate of distribution of drug after its absorption? So there are 2 main points we should keep in mind. This is lipid solubility. This is one of the most important parameter of any drug, and the second one is ionization, and other are blood flow to the binding of the plasma protein site, and plasma protein binding along with the cell protein binding. These are important factors which may alter the redistribution rate. So let me explain you all this point in a very short manner. So first of all, lipid solubility. If the drug is lipid soluble or if the drug is highly lipid soluble drug, then it may distribute throughout the body, including those three barrier: blood-brain barrier, blood-placental barrier, and blood-testis barrier. But if the drug is water soluble, or we can say lipid phobic or lipophobic, that means this drug is water soluble, not lipid soluble drug. So this drug will able to cross only those barriers which allow the molecules of the drug which is having aqueous pore, like in case of absorption, but that drug will not be able to cross blood-brain barrier and these two other BPB and BTB barrier. In case of ionization, you know the point about ionization. When the drug is in unionized form, that drug can easily move through one part to the other part. But once the drug get ionized, the drug will behave like a macromolecule, means large size molecule, and that will not be able to absorb as compared to the unionized form. So that is important. And if the absorption is not adequate, how can distribution will be adequate? So all these parameters of distribution are directly or indirectly somehow related with the absorption parameter. The next one is blood flow. Suppose in my body part, just for example, let me give you a few basic examples which are not actually in taking place. So just for example, my head portion is receiving much amount of blood, but while on the leg side is about comparatively very less amount of blood is going through, passing through, circulating through. So what will happen? If the blood is higher on my brain size, head size, so the drug which is dissolved in the blood will reach to the brain much in quantity as compared to the food side. But just for example, so the rate of blood flow to the particular organ or tissue is very determining step. We can say rate limiting step or major step for the distribution point of view. Higher the blood circulation, higher the availability, higher the distribution of drug to particular organ or particular site of action where it has to be act accordingly. Next one is binding to plasma protein. This is a limiting step. What is the meaning of binding to plasma protein? Suppose I am giving any drug and this drug is having property to bind with the plasma protein and somehow with the cellular protein. So the plasma protein will bind with this drug and keep that drug with itself, and that drug will not be available for the distribution. So if the drug is not available for the distribution, how that particular drug will produce their effect? As in case of chocolate, I have explained you that I have kept 5 chocolate with me. I am not releasing this 5 chocolate for the circulation. So how the remaining 5 students will get this chocolate? Until I will release one by one or simultaneously or at a time, they will have only then they will get the chocolate if I am releasing that thing. So in case of plasma protein binding, the plasma of specific condition bind that drug which is having property to bind with the plasma and retain itself, and that will not be available in the circulation. Same like cellular protein can also bind few amount of drug and that will not release for their action. But after some time, when there is a gap in a saturation of drug in our body or the drug is get eliminated, the concentration of drug in body will be reduced, then only this plasma or drug binding will be disassociated and that plasma protein will release the drug for the absorption. So this is an important factor. This may convert into accumulation effect, and that will be our next topic of in upcoming lecture video. So this is very important. You should keep all points regarding plasma protein binding very carefully. The next slide I am going to explain this plasma protein binding in a specific manner exclusively to plasma protein binding. Now we should take a note that is very important. Unionized lipid soluble drug have widely distribution rate. As I have explained you in the point, only those drugs which are unionized or which are lipid or highly lipid soluble drug can distribute it throughout the body in a good manner, widely distribution. So now we have to understand the basic terms regarding plasma protein binding, and the remaining part will be continued in the next whiteboard section of the same video. So what is the meaning of plasma protein binding? Just simply cut this word into meaningful sentence. We have plasma, protein, and the binding. What is a plasma? Is a constituent of blood. What is a protein? Is a major constituent of the cell. And what is a binding? That means occupancy, that will take part on that particular process, that will take a seat on that particular process. So whenever we give the drug, according to the nature and property of the drug, few drug depending upon their acidic nature or basic nature, they will get binded with the different types of plasma protein. And if the drug is bounded with the plasma protein drug, that drug is not free for the movement. And if the drug is not free, how that drug can produce their effect? That is called plasma protein binding. That means if the drug is having higher plasma protein binding drug, their availability will be less and their effect will be less for the particular time interval. Now let's see the important phenomena and process under the plasma protein binding. First point say, drug, whenever we take drug of any route, from any route of drug administration, that drug, any route of drug administration which reach the circulation, if the drug is given through oral route, that will go through the disintegration, dissolution and absorption, then finally circulation. But if the drug is given directly via the intramuscular route, that will directly reach to the circulation. So after the circulation, drug is having chances, or drug is having property depending on the drug to drug, nature to nature of drug, that will bind to the plasma protein. And what will happen after plasma protein? What are the specific criteria of the drug for the binding of plasma protein? So two different types of drug are there according to their nature of acidity or basicity. Acidic drug and basic drug. Both acidic and basic drug will not be binded with the same plasma protein. There are two different types of plasma protein which may bind acidic drug and which may bind basic drug. So what are these specifications? These are very important for your GPAT, pharmacist, sniper, drug inspector, hospital pharmacist or any other type of pharmacist examination. So please keep this point very clearly in your mind for the longer duration of time, so it will be helpful for you. So acidic drug, those drugs which are acidic in nature, will bind to albumin. One of the most important question which has been asked in so many times. So they will be bind to the albumin, whereas the drug which is having basic property will have opportunity to bind with the alpha acidic glycoprotein. You should remember these two terms. Now let me give you the example regarding this binding site with the help of diagrammatic representation. So there are two types of drug. In case of blue circle, this is representing the acidic drug, whereas the red one is representing basic drug. So same as like that, that is a binding site looking like U shape. This is albumin, which is a binding, it is a plasma protein which occupied by the acidic drug, whereas this is looking like receptor. These are alpha acid glycoprotein and it is binding site for the basic drug. So after available this drug in the bloodstream, what is going to be happen? They will going to occupy their respective binding site, or we can say binding of the drug with specific reference to types of plasma protein. So the blue one is acidic drug and this blue U shape is for albumin. So this acidic drug is going to bind in the albumin, whereas this red circle which is representing basic drug and this structure like shape like of structure is representing alpha acid glycoprotein. So basic drug will occupy in this alpha acid glycoprotein. So these are two specific binding phenomena. This is acidic drug plasma protein binding, this is basic drug plasma protein binding, and there is one possibilities. Acidic drug took place in the respective binding site, basic drug took place in respective binding site, and the third possibility is that few drug will be remain as such unbinded with the plasma protein. These are called free or unbounded drug. This is our interest. This is the point of interest, I must say. These drugs are free for the action. So if you are having 10 number of drug given in the blood, and out of 10 number of drug, few, that means 2 binded with the acidic and 2 binded with the basic, so 4 has been occupied with the plasma protein. We have remaining 6 drug unoccupied or free or unbounded drug. So we will have action just because of this 6 drug, and we will not be able to take action of this 4 drug until this plasma protein and drug binding will disassociate. So once they will dissociate, the drug action will be add up. One by one they will be getting additive effect. So this is important, and that may cause additive effect, or we can say cumulative effect too. That will be discussed in the later classes. So next point say acidic drug, basic drug will respectively bind with the respective albumin and alpha acid glycoprotein. So only free or unbounded drugs are available for their action along with their absorption, metabolism and elimination. Only unbound drug will be produced this kind of pharmacokinetic profile. Now what is the basic example of this drug which may affect the plasma protein binding? Because not all the drugs are same in respect to plasma protein binding. Few drugs are there which are totally bounded with the plasma protein, whereas there are some drug also which are not bounded by the plasma protein. So these are very important example. Example is warfarin, anticoagulant drug. This warfarin is binded 99% with the plasma protein binding. That means only 1% of warfarin is available for their action, whereas the lithium is 0% binded with the plasma protein binding. That means it is completely available for the action. So as you can see with this example, there are drastic difference between these two drugs. From one is the bottom to the top differences. This is only 1% available for the action, and that drug is 100% available for their pharmacological action. Pharmacological action also include the unwanted effect. So you should keep this thing in mind also. Now, what are the basic example of these other plasma protein binding drugs? So see, highly protein bounded drugs which are having property to get binded with the plasma protein are warfarin, furosemide, diazepam, indomethacin, tolbutamide, phenytoin, sulfonamide and salicylate. These are the example of highly plasma protein bounded drug. So this was the first whiteboard section regarding distribution and plasma protein binding. Allow me to move to the second whiteboard section so that I will continue the remaining point regarding plasma protein binding. So let's come with me to watch the next part. So now we are going to study about the clinical significance of plasma protein binding of the drug which has been prescribed by the prescriber or which has been taken by the patient. What is significance? That means clinical significance of plasma protein binding. As we have studied in the previous part, plasma protein binding is one of the most important factor which may affect the distribution of the drug. So whenever we take any of the drug via any of the route of drug administration, either it may oral or somehow parenteral, the drug will go for the absorption in systemic circulation. But after absorption, the drug should available throughout the tissue in our body, and for that the drug should distribute equally. And this factor, that means plasma protein binding factor, can affect that distribution process. If the drug is acidic, the drug will bind to the albumin protein. If the drug is basic, the drug will bind to the alpha acid glycoprotein. But only those drugs which are free, which are not bounded with the either acid or basic protein, plasma protein, will produce their pharmacological action, and this is first point of our presentation. See, only free fraction of drug is available for their pharmacological action, their metabolism and their excretion. Those drugs which are bounded by plasma protein will not be able to produce their pharmacological effect. Pharmacological effect include both desirable and undesirable effect. So only fraction which is free from the plasma binding will produce their pharmacological action, and that is the importance of our considerations. Next point say, when free drug fall in the body, then the bounded drug is released. So only those drugs which are free will produce their effect, but the free drug will be get metabolized, and after some time the drug which is free now get eliminated from our body. So what is happening now? What is going to be happen now right now actually? So those drugs which are bounded by plasma protein will now slowly released by that plasma protein, and that bounded drug become free drug. Now that free drug will produce their pharmacological action. Again they will go for metabolism, and finally they will go for the excretion. So until the free fraction of drug is available, the plasma protein binding will not be released. This is important factor. Now next point say, this is a very important one, protein binding serve as a store or reservoir. What is the meaning of that? See, if you have much amount of money, you don't need any extra, so you put some of them into reserve cases, so that in case of emergency you may utilize that money. But in this case of drug plasma protein binding, if the drug is bounded with the plasma protein either acidic or basic, the drug will be stored. The drug will be act as a reservoir for the drug. That means that is not available for the action right now. So whenever there will be need, that means whenever the free drug get out of the body, that reservoir or that store will start releasing the drug, will start unbounding the drug from the plasma protein binding. So till that is acting as a store or reservoir. So another important point is here in the fourth number, the protein binding is very important. Protein binding phenomena increase the half-life of the drug. This is drug specific, and if the drug half-life will increase, their duration of action will also be increased, and their drug will be a long-acting drug. So this is important. 3 points are included in this point. Plasma protein binding can increase the half-life. What is the half-life? The half-life is a life at which the concentration, also the initial concentration of the drug, remain half of its initial concentration or get eliminated of its hard portion from its initial concentration. Either it is present in the plasma or either it is removed from the blood or body at its half concentration is known half-life. And the duration of action is the time or maximum time till the effect of drug persists in our body is called duration of action. So that two factor can be governed by this plasma protein binding. Now coming to the next one, drug may fight for or drug may compete for the same binding site if the drug is more higher affinity will displace the another, that may result into drug interaction. See, the plasma protein binding can be of two types, for acidic and basic drug. But in case of this, for example, in case of acidic drug are there, but two drugs are of different affinity for the particular plasma protein binding, that is albumin. So if one drug, that is A drug and B drug, but if the A drug is having higher affinity for that particular albumin protein, then it may replace or displace the B drug from plasma protein binding. So that phenomena, that is called competitive inhibitor phenomena, may affect the drug action which has been taken initially or in starting phase, that can be converted or result into drug interaction. So in the future case, whenever you will give such kind of drug, that may affect or that may displace the one drug by the other drug, and that is very important phenomena for the unwanted effect point of view or cumulation effect point of view. To make this concept better understand by you, I am giving you the example. So if the example is warfarin, because in the last slide we have studied about the warfarin. This is one of the most important drug which is binded 99% with the plasma protein. What is the meaning of that? If I am giving 100% of warfarin, out of 100%, 99% of the warfarin will be binded with the plasma protein. Only 1% of warfarin will be free, and that will produce the pharmacological effect. But in case of those situations where drug interaction is taking place, I am giving warfarin and I am giving endomethacin simultaneously, then what will be happening, about to happen, this endomethacin will reduce that means reduce the plasma protein binding of warfarin up to 95%. See, in initial level warfarin was bounded 99%, but just because of the influence of endomethacin, this binding ratio has fallen down to the 95%. So what will going to the free drug availability? In initial level that was only 1% free for the action, but now this has been changed and now this is 5% available for the action or pharmacological action. So decrease in the plasma protein binding, or we can say increase in the free drug concentration took place for warfarin just because of endomethacin. So this is called drug-drug interaction, because all those two things, warfarin and endomethacin, both are drug. So this is called drug interaction. Now coming to the next point, protein binding site may get saturated with repeated administration of drug. So more drug remain in the free form. If you repeat the drug, or if you take that drug, same drug in a frequent interval, so that the plasma protein binding site will be get saturated. So now afterwards there will be no plasma protein binding or less plasma protein binding for that particular drug in case or in comparison with the previous one. So now what's going to be happen? If the drug is not bounded to plasma protein, that drug will be remain as such or remain free or remain unbound. So you know it very well that only unbound or free drug is available for the action. So if the drug is not bounded with the plasma protein, more drug will be available for their pharmacological action, and that free form of drug may produce higher response as than usual one, and that may convert it into a toxic or accumulative effect. That is important one. So there are two more important phenomena regarding this: the chronic liver or renal failure. In case of those patients who is having chronic liver failure or renal failure, that means kidney failure, what is going to happen? In such patient there is a condition called hypoalbuminuria, that means reduction in the amount of albumin. So you know it very well, albumin is a plasma protein binding site for the acidic drug. So if in this condition of renal failure and kidney failure, the level of albumin is reduction, or there is reducing the level of albumin, so what is going to happen? How the plasma protein binding will take place for the acidic drug? Because level of amount of albumin is getting low, so there will be an increase in the free form of the drug, and if the drug free form is available in the blood, that may produce toxicity, because you have not expected this unwanted or much amount of the drug in a free form. So last point, say if the person is having inflammation condition, it is respect to the basic drug. So if he or she is having inflammation, then this inflammation increase the chances or availability of alpha acid glycoprotein, which is a plasma protein, which is a binding site for basic drug. So much amount of drug will get bounded to the plasma protein, and less amount of drug will be available for their pharmacological action. So there the action may be reduced. So in both of the case you can see the opposite response. In this case albumin is getting low, so free drug is getting increases. In case of this, alpha acid glycoprotein are getting increasing, so the plasma protein binding is increasing, the free drug is reducing. So though both are opposite to each other. So this was simple but important concept regarding distribution and the plasma protein binding. So that's all for today. Thank you very much for watching this video. The reference for today's topic is KD Tripathi and P. Jagdish Prasad. The edition of KD Tripathi is 8th, that is the latest edition. So friend, that's all for today. Thank you very much for watching this video. The reference I have told you is very important. If you do not have that book, then not to worry about it. You can have the screenshot of both whiteboard section after some time. So please do take care of yourself. Thank you.